All right, hey everyone. Welcome to another deep dive. This time, we're going to get you prepped for that pharmaceutical biotechnology exam, really understanding those antimicrobial agents.
Exactly. Think of this as your super focused study session. We're going to break it all down, how they work, how bacteria resist them, even how this all applies to treating infections and developing new therapies, like even cancer
Yeah,
therapies.
we've got your notes, all the diagrams, those complex pathways. But before we get too deep, let's start with the basics. What even is an antimicrobial agent?
Well, they're basically our molecular weapons against those tiny invaders, right? Bacteria, fungi, viruses, even parasites. Some are natural, like penicillin, and some are made in the lab. The key is they can kill those microorganisms or at least stop them from multiplying.
Okay, that makes sense. But why is it so important to really understand those mechanisms of action? Is it more than just, memorizing names and structures for the exam?
Oh, absolutely. Imagine a doctor trying to treat an infection without knowing which antibiotic targets which process, right? It'd be like just throwing darts in the dark. Understanding the how allows for better, more targeted treatment and helps us develop new therapies as those microbes evolve and become resistant.
Got it. So it's about precision in treatment and staying ahead of the microbes.
Hmm.
Our notes have a section called cell wall attackers, which sounds pretty intense. Where do we start with these?
So think of a bacterial cell like a fortress. The cell wall is its outer barrier. If you can break down that wall, the cell can't survive.
Okay, so some antimicrobials actually target this vulnerability.
Exactly. A classic example is the beta-lactam family, with penicillin, being the most famous one. They bind to these proteins, PBPs, which are enzymes that are key for the final steps of peptidoglycan synthesis.
And peptidoglycan is what gives the cell wall its strength, right?
Precisely. Beta-lactams stop that process, weaken the cell wall, and then boom, the bacterium bursts open. It can't survive the internal pressure.
So the bacteria basically explodes because its wall can't hold up. Are there other agents that attack the cell wall in different ways?
Definitely. There are the glycopeptides like vancomycin. Imagine those peptidyl glycan building blocks as bricks. Vancomycin just grabs onto those bricks, preventing them from being used to build the peptidoglycan chain.
So it's like sabotaging the construction by intercepting the building materials. I see why understanding these mechanisms is so important for developing new drugs.
Absolutely. And then we've got bacitracin, which takes a whole other approach. It blocks the transport of those building blocks across the cell membrane.
Like a roadblock, preventing the components from getting to the construction site. So we've got the direct attack on the builders with beta-lactams, them messing with the bricks themselves with glycopeptides, and now we've got the transportation shutdown with bacitracin.
Yeah, a great way to put it. Don't forget, your notes have some great diagrams showing these processes. Connecting those visuals to what we're talking about will really help it stick.
Yeah, visuals are key for sure. They make these complicated processes much easier to understand. But I've got to wonder, if bacteria are under attack all the time, they must have some defenses, right?
You're absolutely right, and that's where things get really interesting and a bit concerning. That's where we get into the whole world of antimicrobial resistance, which we'll dive into later. But for now, let's move on to another target for these antimicrobial agents: protein synthesis.
Protein synthesis. That's the process that keeps every cell alive, right? Bacteria, too. We covered that in basic biology. How does it become a target for these agents?
Well, it's all about exploiting the differences between bacterial and human ribosomes. Remember, ribosomes are like the factories that build proteins using genetic instructions.
So by targeting bacterial ribosomes specifically, we can disrupt their protein production without hurting our own cells.
Exactly. One group, the aminoglycosides, they bind to the 30S subunit of the bacterial ribosome. This basically causes the ribosome to misread the genetic code, leading to faulty proteins.
Like introducing a glitch into their assembly line, causing chaos in their cellular processes.
That's a great way to think about it. Then you have the tetracyclines, which act like bouncers at the ribosome's entrance. They also bind to the 30S subunit, but instead of messing with the code, they block tRNA molecules carrying amino acids from attaching.
So no supplies, no proteins.
Exactly. And then we have the macrolides and the lincosamides. These target the 50S subunit and interfere with the movement of the ribosome along the mRNA.
So instead of messing with the message or the supplies, these are like jamming the machinery itself, preventing the smooth production of proteins.
Yeah. And we haven't even talked about chloramphenicol or the oxazolidinones, which have their own unique ways of messing with protein synthesis.
Wow, there are so many different ways to disrupt this one process. It's clear that understanding these mechanisms is key, not just for exams, but for appreciating just how complex this fight really is. But bacteria are adaptable, right? They're not just going to sit back and let us dismantle their factories.
Of course not. And that brings us right back to antimicrobial resistance. But before we get into how bacteria fight back, let's talk about another target. DNA and RNA. They're very core.
Ooh, now we're getting to the genetic level. Things are getting really interesting. Tell me about those nucleic acid saboteurs, as our notes call them. How do they disrupt the very essence of bacterial survival?
All right, welcome back. So last time we were talking about how those antimicrobial agents target the cell wall and protein synthesis, the bacterial strongholds.
It's pretty amazing all the different ways scientists have found to take them down, disrupt those essential processes.
Yeah, but as we mentioned, the bacteria aren't just giving up. They've got their own defenses, which brings us to antimicrobial resistance.
Okay, so let's break that down a bit. What does antimicrobial resistance actually mean, like practically?
Imagine a drug that used to work great, suddenly doesn't. The bacteria have figured out how to survive the attack, makes our treatments useless.
So it's like those molecular warriors are losing their edge against a tougher enemy. What makes bacteria so good at developing this resistance?
Well, a few things. First, they multiply really fast, sometimes like every 20 minutes. That plus their simple genetic makeup means mutations, changes can pop up and spread through the population super quickly.
So it's like a constant game of chance with these mutations appearing.
Mm.
And some of them just happen to give the bacteria an advantage when a drug is around.
Exactly. And because they reproduce so fast, those resistant ones can become the majority in no time. Plus, bacteria can share genetic information not just with their offspring, but also with each other, like directly.
Wait, so they can like trade resistance genes like trading cards? That's wild.
It is. They do it through different ways, like conjugation, transformation, and transduction. So resistance spreads rapidly, even between different species. It's like they're sharing battle plans, helping each other outsmart us.
So we're not just dealing with like one type of bacteria becoming resistant. It's more like this huge network sharing and boosting their defenses. That's a bit daunting, isn't it?
It really shows how complex this challenge is. And remember, resistance isn't always something they develop. Some bacteria are just naturally resistant to certain drugs, like mycoplasma. They don't even have a cell wall, so drugs targeting peptidoglycan synthesis, like penicillin, are totally useless.
Like trying to, I don't know, use a sword against a ghost, that target. But going back to acquired resistance, our notes talk about different mechanisms. Can we go over those?
Absolutely. One common one is enzyme inactivation. Bacteria can produce enzymes that basically break down the drug, make it useless. A classic example is beta-lactamases. They target those beta-lactam antibiotics we talked about. They break the beta-lactam ring, which is what makes those drugs work.
So it's like having these little saboteurs inside, destroying our weapons before they can even reach their target. Are there other enzyme saboteurs like that?
Tons. Bacteria can also modify aminoglycosides, another important group of antibiotics. They add chemical groups, changing the drug's shape so it can't bind to its target on the ribosome.
Like putting a disguise on the drug so it's not recognized. Smart, but also kind of annoying. What other tactics do they use?
They can modify the drug's target site, making it harder for the drug to bind and do its job, like changing the locks on a door. For example, mutations in the genes for ribosomal proteins can change the binding site for macrolides, making them less effective.
It's this constant back and forth. We develop new drugs, and they find ways to make them obsolete. Feels like a never-ending cycle.
It is an arms race, yeah. And then there's cross-resistance, where resistance to one drug can make them resistant to others in the same class, even if they've never encountered them before, because those drugs often have similar structures or target similar pathways.
So resistance to one drug can protect them against a whole family of drugs. That's a huge setback.
It is. And then the biggest challenge, multi-drug resistance, or MDR. Those bacteria are resistant to multiple classes of drugs, making treatment incredibly difficult.
That's scary. What causes that? Is it just like a bunch of different resistance mechanisms all combined?
Often it's a combination of things. We've talked about enzyme inactivation and target site modification, but another big one is efflux pumps. They're protein pumps in the bacterial membrane that act like little vacuum cleaners, sucking the drug out of the cell before it can do anything.
So they're literally pumping the drug out. That's so efficient. What powers these pumps?
It depends on the pump. Some use proton gradients, others use ATP hydrolysis. The point is, they're really good at keeping the drug concentration low inside the cell, making it useless.
It's like they have a pump for everything, ready to spit out whatever we throw at them. Are there any other ways they avoid our attacks?
Oh, yeah. Some bacteria can reduce their permeability, making it harder for the drug to even get in. They change their porin channels, those little gates in the cell membrane that control what goes in and out. Really effective against hydrophilic antibiotics like beta-lactams, which need those channels to enter.
So it's like they're building thicker walls, making it even harder to get through. But I have a feeling there are even more tricks, aren't there?
Of course, some bacteria have bypass mechanisms so they completely avoid the drug's effects. For example, some bacteria can overproduce the enzyme that an antibiotic targets, basically flooding the system with it.
So they overwhelm it with sheer numbers, diluting the drug's impact. Clever, but frustrating. Any other examples of those bypass mechanisms?
Yeah, some bacteria develop alternative metabolic pathways, like finding detours around the roadblock. They use different enzymes or even completely different reactions to achieve the same goals.
It's crazy how adaptable they are, always evolving and finding ways to survive.
It's natural selection at work. The bacteria that survive our attacks are the ones that pass on their resistance genes, shaping the next generation. That's why understanding these resistance mechanisms is so important. It helps us develop new strategies to fight back.
This has been a fascinating, if a little scary, look into antimicrobial resistance. It's clearly a constant battle, and we need to understand the enemy's tactics to stay ahead. But I'm curious, how do antibiotics affect us on a larger scale, beyond just fighting infections?
That's a great question. Antibiotics have changed healthcare, agriculture, even scientific research. We can talk about those fascinating applications next time.
Sounds good. Stay tuned, everyone, as we explore the wide-ranging impact of these tiny but powerful molecules. Okay, so welcome back to our deep dive into antimicrobial agents. We've talked about how they work, those different mechanisms, and also that pretty serious issue of antimicrobial resistance.
It's a really complex field, and it's changing all the time, but these are the fundamentals, right? Anyone going into biotech needs to understand this stuff.
For sure. So let's zoom out a bit from the microscopic level and talk about the bigger picture. Antibiotics, they do more than just fight infections in a lab, right?
Oh, yeah, definitely. Antibiotics have totally changed how we approach healthcare, agriculture, even scientific research. They're everywhere, impacting us in ways we might not even think about.
Okay, so let's start with the most obvious one, medical treatment. It's hard to imagine medicine without antibiotics.
Right. Before penicillin, even small infections could kill you. Now we've got all these different antibiotics to treat bacterial infections, from like simple UTIs to really serious stuff like pneumonia.
It's amazing how many lives have been saved because of these, I guess you could call them molecular warriors.
Yeah.
But it's not just about treating existing infections, right? Aren't they also used to prevent them?
Exactly. Prophylactic use is super important in certain situations, like for patients having surgery. They often get antibiotics beforehand to prevent infections, and people with weak immune systems or those at risk for specific infections, they might get them too.
So it's like giving them a shield, just in case, to prevent complications. But I'm guessing that using antibiotics so much, even preventatively, is probably contributing to that whole resistance problem we talked about.
Yeah, it's a tough balance. Prophylactic use is important, but we have to be careful, use them only when we really need to. Overusing them definitely speeds up the development of resistance.
Speaking of widespread use, antibiotics aren't just for humans, right? They're also really important in veterinary medicine.
Absolutely. Just like in human medicine, antibiotics are key for treating infections in animals. Everything from like respiratory infections in cows to skin infections in, our pets.
Good to know our furry friends can benefit from these treatments too. But I've heard there's like a bit of controversy around using antibiotics in livestock. What's that about?
It's about using antibiotics for growth promotion. For years, farmers have been adding low doses of antibiotics to animal feed, not to treat infections, but to make the animals grow faster and produce more.
Wait, so antibiotics can make them grow faster? How does that even work?
We're still figuring out the exact mechanisms, but it seems like low doses of antibiotics might change the gut microbiome, which helps them absorb nutrients better. The problem is, this practice has been linked to antibiotic resistance.
So the worry is that we're basically creating a ton of resistant bacteria in these animals, and those bacteria can then spread to humans through the food we eat.
Exactly. We could be creating this huge reservoir of resistance genes that could make some of our most important antibiotics stop working, not just in animals, but in humans too. It's a big public health issue, and a lot of countries are putting in stricter rules to limit the use of antibiotics for growth promotion.
Yeah, it's a complicated issue for sure, balancing, food production with keeping people safe.
Yeah.
So antibiotics are used in medicine, they're used in agriculture, but they're also used for food preservation, right?
Right. They're used in different ways to keep our food from spoiling and make sure it's safe to eat. For instance, niacin, a naturally produced antibiotic, is added to a lot of dairy products to prevent the growth of Listeria monocytogenes, a bacterium that can make you really sick.
So they're even working behind the scenes to keep our food supply safe. They really are versatile, these little molecules.
And it doesn't stop there. Antibiotics have become super useful in scientific research, especially in molecular biology.
Oh, really? How so? How are they used in research?
Well, certain antibiotics can block very specific biological processes, which lets researchers study them more closely. For example, rifampicin is often used to study RNA synthesis because it targets bacterial RNA polymerase.
So it's like using a tiny tool to stop one specific part of the cellular machinery so scientists can see what happens and learn more about how it works.
Exactly. And tetracyclines are used in genetic studies to look at gene expression. They can actually control which genes are turned on or off, like little switches.
It's amazing how these molecules, which were discovered because they could fight infections, are now used in so many different ways.
Yeah, that's the beauty of science, always asking questions and making connections. Discoveries in one area can lead to unexpected applications in others.
Well, we've covered a lot today, huh? We've really explored the world of antimicrobial agents, how they work, the challenge of resistance, and all the different ways they impact our lives.
And for anyone interested in biotechnology, understanding all of this is so important. It's not just about memorizing facts, it's about grasping how complex this field really is and contributing to the future, developing new therapies, strategies to fight resistance, and innovative ways to use these molecules.
This deep dive has been really eye-opening, that's for sure. I feel like we have a much better understanding of these tiny but powerful molecules.
Remember, there's so much more to learn about microbes. Never stop being curious, asking questions, and pushing the boundaries of what we know.
Who knows, maybe one of you listening will be the one to discover the next big antibiotic or find a totally new way to use these molecular warriors.
Keep learning, keep exploring, and keep diving deep into the world of science.